A very pleasant and delightful morning to everyone present over here. Honorable Director of Roland Institute of Pharmaceutical Sciences, Dr. J. Surya Rao sir, respected Chairperson Srimati J. Jayalakshmi Madam, Principal as well as Coordinator of today's program, Dr. B.V.V. Ravi Kumar sir, Vice Principal Dr. C.H. Niranjan Patro sir, Co-Coordinator of this event, Dr. Viswa Mohan Sahu sir, today's chief guest, Professor Prabhat Kumar Sahu sir, guest of honor of today's program, Professor Sabu Thomas sir, eminent speakers, delegates from all over the country, my dear colleagues, and all the great minds present here. I, Ms. Deepika Rani Sahu, Assistant Professor, on behalf of RIPS, would like to present a gracious welcome to you all to this inaugural ceremony of Series 3 Online Faculty Development Program on Emerging Trends in Drug Design and Development, sponsored by All India Council for Technical Education, New Delhi, and organized by Roland Institute of Pharmaceutical Sciences, Odisha. Before starting the inauguration, let me focus some few highlighting points about this FDP. The major focus of conducting this FDP is to train the faculty members in the field of pharmaceutical education and research. The main objective of this FDP is to update all of us about the new technologies applied during drug discovery and development process in pharmaceutical research. Drug design is the key step to generate new drug molecules in drug development process. So this program will help all the faculties to improve their innovative ideas of their research works. It is going to develop technical skills of young faculties while designing new laboratory experiments, project works for students. This program is also going to give an insight into field by discussion of necessary theoretical background as well as live interaction with scientists, academic resource persons, experts from different industries and research laboratories. Now, to mark this event a traditional beginning, let us begin a virtual presentation of lamp lighting followed by Sri Ganesh Vandana.
शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्नमोऽस्तु ते शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः
वक्रतुण्डमहाकाय सूर्यकोटिसमप्रभा निर्विघ्नं कुरु मे देव सर्वकार्येषु सर्वदा सर्वकार्येषु सर्वदा सर्वकार्येषु सर्वदा
Now it's time to introduce our institute by Viswa Mohan Sahu sir. Sir, Now it's time to invite our honorable patrons, Srimati J. Jayalakshmi Madam, Chairperson, RIPS, and Director Dr. J. Surya Rao Sir on board for the welcome address. Over to you, Madam.
Namaste. Most respected Chief Guest on this occasion, Professor Prabhat Kumar Sahu, Director DIPSAR, the Constituent College of Delhi Pharmaceutical Sciences and Research University, and also the ex-director AICTE. Today's guest of honor, Professor Sabu Thomas, the Honorable Vice Chancellor, Gandhi University, Kottayam, Kerala. Respected Principal Sir, Professor Dr. BBB Ravikumar, Vice Principal Professor Niranjan Patro, and the co-coordinator for today's program, Dr. Biswamohan Sahu, other faculty members, supporting staff, participants, delegates. Once again, good morning to all of you. At the very outset, I am very much happy, delighted, and to see the most vibrant guests on this platform as the chief guest and guest of honor. Really, we are very much privileged and honored, sir. And on behalf of RIPS, I extend my heartfelt thanks to both of you because you have accepted our invitation and be our guests on this platform where we could able to see your faces on this computer screen. You are, sir, the powerhouse of knowledge and the dynamo of executing the things in pharmacy world. I can assure and reassure and challenge that such kind of people we wanted to see on this platform, and feeling very proudly that the befitting guests we have today on this webinar. So during this pandemic situation, there is no other option to see each other, to meet each other, to exchange our thoughts, everything on this webinar only, on this online mode only. Sir, regarding this RIPS, this is 35 years old college, and since from beginning till today, our focus is only to provide quality education. So quality is always our passion, and we train and retrain our teachers, including our principal sir, to have a fine tuning in giving the academics in their best own way. Therefore, we never compromise with the quality, and thereby RIPS had a special name and fame in the state of Odisha, not only in the state of Odisha but also in the nationwide. As our chief guest was sharing his thought now, that RIPS had its name throughout the nation. So we are very much privileged and honored to receive such kind of compliments from such great people like you, sir. In our institute, we focus always equally that on research and academics. In an educational institute, the research and academics are the two eyes. So we should not neglect, and we should not ignore, and we should justify always in every platform. Thereby, we could able to organize such kind of programs, and always our thinking is all that what best we can give to our students beyond our syllabus. So that is our focus. Thereby, we are applying for different projects, and AICTE is kind enough providing this kind of project, that is faculty development program. And thanks to AICTE for their funding, for their support to organize this program. And very successfully we could complete the series 1 of faculty development program as well as the series 2 with the support and cooperation of so many speakers, so many resource persons, not only nationally but also internationally. So we could able to blend all these things very nicely. Thanks to coordinator sir, our honorable principal sir. Thanks to co-coordinator Dr. Biswamohan sir for arranging such a very galaxy of wonderful resource persons for this on this August platform. I hope that this series 3 will also take a very very successful way, and hope that all these participants, delegates will definitely enjoy. And this is not only a program for faculty development, this will be helpful for the students, faculties, and also researchers. Thereby, once again, I thank the organizers for giving me this opportunity to welcome our very most dignified guests today, Professor Prabhat Kumar Sahu sir, and also Professor Sabu Thomas sir, for accepting our invitation and to be our chief guest of honor today. So very 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 nice to you, and hope that all will definitely enjoy this series 3 equally. So thank you organizers for giving me this opportunity. With this, I welcome all my again the guests, the faculties, the students, and also the participants, delegates to this online program of faculty development program of series 3. Thank you.
We cannot hear you.
Deepika ma'am, it's not audible.
Sorry, madam.
We cannot hear you.
Is it audible, sir, now?
Now, okay.
Yeah.
Thank you so much, madam, for your constant support and encouragement. Now I take this opportunity to invite our respected principal, Professor Dr. VVV Ravi Kumar sir, coordinator of this FDP, to address the gathering.
Thank you, Deepika. Chief guest of today's program, respected Professor Prabhat Kumar Sahu sir, Director, TIPSR, University of Delhi. Guest of honor, respected Professor Sabu Thomas sir, Honorable Vice Chancellor and Professor, School of Energy Materials, Mahatma Gandhi University, Kerala. Patrons of this faculty development program, respected Mrs. J. Jayalakshmi Madam, Chairperson, Roland Institute of Pharmaceutical Sciences, and Dr. J. Surya Rao sir, Director, RIPS, Vice Principal Professor Niranjan Patro, Co-Coordinator for this program, Professor Visumohan Sahu, staff members of Roland Institute of Pharmaceutical Sciences, and my dear delegates who registered for this faculty development program. A very good morning to all of you. At the outset, I welcome all of you to this inaugural function of the 2 weeks faculty development program, Series 3, sponsored by All India Council for Technical Education, New Delhi, which will be held from 19th April 2021 to 30th April 2021. I'm very happy to inform all of you that we have completed Series 1 and Series 2 successfully in the months of February and March. And for this series also, we have received good response from faculty members all across India. I can promise you that this program will definitely help you to improve your knowledge and skills in the area of drug design and development, as this program is lined up with lectures and demonstrations by reputed resource persons from not only India but also from abroad. I'm highly thankful to all the resource persons who agreed readily to share their knowledge and wisdom with all of us. I take this opportunity to appreciate Dr. Visumohan Sahu, who have taken all the pain to make arrangements to conduct this FDP almost single-handedly. I also thank Vice Principal Professor Niranjan Patro and all the faculty members of RIPS for their support. I thank management of Roland Institute of Pharmaceutical Sciences for their support and guidance in organizing not only this FDP, and as Madam was telling you, RIPS is a reputed institution, not only in Odisha but also throughout India, for imparting quality education in the field of pharmaceutical sciences. That is only because of uh, the active involvement of the management, I take this opportunity to thank our chairperson, director, and all the governing members of RIPS. I also thank the principals of all the colleges for allowing the teachers to participate in this FTP. And I thank AICTE for funding this program. And my sincere and heartfelt thanks to today's guests, Professor Prabhat Kumar Sahu sir and Professor Sabu Thomas sir, for accepting our invitation and gracing this occasion to motivate all of us in spite of their busy schedule. With this, I once again welcome all of you to this third series of online faculty development program sponsored by All India Council for Technical Education, and wish this program will be conducted successfully and meaningfully. Thank you.
Thank you so much, sir, for your all-time support and constant monitoring. I will now invite our respected Vice Principal, Professor Dr. C.H. Niranjan Patra, sir, for sharing his views about this FDP. Over
Good
to you,
morning,
sir.
all. Am I audible?
Yes, sir, audible.
So, esteemed Chief Guest Professor Prabhat Kumar Sahu, Director DIPSR, New Delhi, esteemed Guest of Honor, Professor Sabu Thomas, Vice Chancellor, School of Energy Materials, Mahatma Gandhi University, Kerala, other dignitaries present in this online platform, and my dear participants, very good morning and warm greetings from Roland Institute of Pharmaceutical Sciences, Berhampur. I would like to address the gathering with a quotation of Swami Vivekananda, and I quote, "Education is the manifestation of the perfection already in man," unquote. So this FDP is all about this. The teachers have the knowledge, they have the talent, but that perfection needs to be nurtured and guided so that they can perform well in their respective institutes and they can perform well in their professional careers, and which in turn will contribute a lot in the nation building process. So I congratulate the organizers of Roland Institute of Pharmaceutical Sciences for taking up this FTP, and my best wishes for the program. Thank you very much for giving me this opportunity.
Thank you so much, sir, for your motivational and inspiring words. Roland Institute of Pharmaceutical Sciences since the year 1986 has been continuously adding success stories to its name, from being one of the best pharmacy colleges in Odisha and have many more tags to its name. Now it's time to hear from our guest of honor, Professor Sabu Thomas, sir. Before inviting Sir for his talk, I will take the opportunity to introduce Sir to all of us. Sir is currently working as Vice Chancellor and Professor of Polymer Science and Engineering, School of Chemical Sciences. He is the founder and director of International and Inter-University Center for Nanoscience and Nanotechnology, Mahatma Gandhi University, Kottayam, Kerala, India. Sir has completed his PhD in engineering from Indian Institute of Technology, Kharagpur in the year of 1987. He has completed BTech in Polymer Science and Rubber Technology from University of Cochin in the year of 1983. Sir has done BSc in Chemistry Honours from University of Kerala in the year of 1980. Sir has worked as Senior Research Fellow in Indian Institute of Technology, Kharagpur from the year 1984 to 1987. After that, worked as lecturer in Mahatma Gandhi University, Kottayam, Kerala up to 1992. From 1992 to 1997, Sir has worked as reader in the same university, and after that worked as regional director in School of Technology and Applied Sciences, Mananam. From 1997 to till now, Sir has worked as professor in School of Chemical Sciences and Director of International and Inter-University Center for Nanoscience and Nanotechnology, Mahatma Gandhi University, Kottayam, Kerala. Sir has won many awards and prizes in his name, like University level postdoctoral fellowship, Fellow of the Royal Society of Chemistry, London, Catholic University Visiting Research Fellowship, Leuven, Belgium, CNRS Research Fellowship, France, Professor Sukumar Maithi National Award for Best Polymer Researchers. He has listed in the panel of most productive researchers in India, fifth position in the year 2008 by National Institute of Science and Technology and Development, New Delhi. He has also awarded with Distinguished Professorship Award, Joseph Stefan Institute, Slovenia in 2010, Fellow of the Royal Society of Chemistry, London, awarded with Bronze Medal of Chemical Research Society of India, Society for Educational and Scientific Research, Nanotech Scientist Award in 2014, and awarded with Dr. APJ Abdul Kalam Award for Scientific Excellence in the year 2016. And many more awards to his credit. His field of research excellences are: Sir has published total 1101 number of articles in various reputed journals. Out of that, hundreds are high indexed Google Scholar and 86 Scopus indexed journals. His total number of citations are 47,634. He has filed three number of granted patents to his credit. He has authored 135 numbers of books, supervised 107 PhD theses. He has received 40 national, international, and industry-supported projects and funds of about 30 crores. He was cited in the list of most productive researchers in India in fifth position in the year of 2008. He was also listed in the most cited researchers in material science and engineering by Elsevier Scopus Data 2016. Sir has visited many countries like Canada, Belgium, France, China, Germany, Slovenia, Strasbourg, Spain, Malaysia, Korea, Poland, Iran, Brazil, Russia, etc. as postdoctoral fellow and visiting researchers professor. He was invited as guest speaker in many universities in Australia, Germany, France, Switzerland, Italy, Netherlands, Belgium, Taiwan, Malaysia, Singapore, Japan, Africa, Europe, Korea, Poland, Iraq, South Africa, Italy, Brazil, Russia, etc. With this brief introduction, I now invite Professor Sabu Thomas Sir on board.
Very good morning. Very good morning to all of you. I'm extremely delighted to be part of this wonderful AICT sponsored 12 days faculty development program on emerging trends in drug design. Professor Sahu, our distinguished guest, Professor Ravi Kumar, the principal, Professor Mohan Bishwas, Professor Rao, Madam Jayalakshmi, all the distinguished speakers for the conference, for this FDP program, and all the participants. Let me first congratulate the Roland Pharmaceutical Institute in organizing 12 days faculty development program. I'm a strong supporter of FDP. I wanted to share an experience with you. I think 5 years ago, I went to the United States as a Fulbright fellow. There were 15 people along with me from India. I represented from Kerala. And we were taken to 12 universities in the US. Let me share an experience when I visited UPenn, University of Pennsylvania in Philadelphia. And I had a meeting with the president of UPenn. UPenn always has a ranking of within 20. It could be QS ranking or Shanghai ranking or Times ranking, whatever be the ranking, they are always within 20 or within 30. I asked the president of UPenn, what is the secret of your success? You know what he said? He said, Dr. Thomas, we have continuous FDP program in the campus. Whenever they get a new faculty in the campus, this faculty is given enormous training. How to write a good research proposal, how to give an effective class, how to value a question paper, how to write a winning project. Additionally, they are given lots of training on emerging areas of science, technology, humanities, social sciences in their domain. Therefore, I strongly believe that faculty development program has a major role to strengthen our teachers. So congratulations to Roland Pharmaceutical Institute in organizing such a beautiful FDP. And I could see the galaxy of people going to speak in this FDP program. You have selected a good panel of speakers. And drug design is a very important topic. Look at, we are facing the corona pandemic now, and the world worked very hard. And within 1 year, we could bring a vaccine. India has a vaccine, US has a vaccine, Europe has a vaccine, China produced a vaccine, Russia has a vaccine. Usually vaccine development, I have colleagues working on drug development in my university. Normally a vaccine development, drug development takes 10, 12 years of time, but this we could do it within 1 year. This is because of cooperative movement. People started working together. People started sharing their know-how. And we could make it within 1 year, a good vaccine. Therefore, I strongly believe that we all have to work together. Industry and university, we have to work together. I mean, if you do that, you can achieve the target very fast. Additionally, people make use of theory, modeling, and simulation quite a lot. Computational approach. So we should use computational approach every field. If you use computational approach in drug design, you can achieve the target pretty fast. Therefore, I request all the attendees, all the participants, to be extremely active in this FDP program. Ask questions, understand the subject, interact with the speakers. Personally, I love conferences, workshops, meetings, symposia. My own domain, nanoscience and nanotechnology. In polymer science, we organize a large number of meetings. I just finished international meetings on nanoscience and nanotechnology just last week. We had 300 speakers, online meeting. Because of this large number of conferences and symposia, you name any country in the world, I have at least 10, 15 academics as my collaborators. Therefore, I request all the participants to be extremely active, interact with speakers, make a very strong network, and we all have to work together. Then we can achieve the results very fast. If you find something interesting in your laboratory, don't publish, you go for a patent. I made a rule in my university that if you find something interesting in the laboratory, go for a patent. Then you publish. Don't stop there. You collaborate with the company and commercialize this. This is the only way we can market our product. And I wish all the best for this wonderful FDP, and I wish good success. And I am also invited to give a lecture. I'm going to give a lecture on scaffolding material, how scaffolding materials could be made, could be fabricated, and could be made use of for drug release applications. I wish all the best for this conference. Thank you so much, and thanks a lot for the kind invitation. Good success. Thank you.
Thank you, sir, for sharing your valuable time with us.
Thank you so much, sir, for sparing your valuable time with all of us and accepting our invitation as well as gracing this FDP with your presence. To the next, we have with us our esteemed chief guest of today's event, Professor Prabhat Kumar Sahu, sir, with us. Before inviting him for his talk, I will take the opportunity to introduce sir to all of us. Sir is currently working as Professor and Head Department of Pharmacy from Delhi Institute of Pharmaceutical Sciences and Research, University of Delhi. Sir has completed his M.Pharm and PhD from Andhra University, MBA from Xavier Institute of Management. He has about 32 years of academic and research experience. He started his career as lecturer in Sri Jayadeva College of Pharmaceutical Sciences, Odisha from 1988 to 1996, then a senior lecturer from 1996 to 1998. After that, worked as reader in Dipsar from 2004 to 7, and then professor in the same institute from 2010 to till date. Sir was worked as former director of AICTE from the year 2010 to 13. He was elected as convener in continuing education program of AICTE, expert committee member, AICTE project evaluation, expert for BIRAC project evaluation, member of visiting committees of PCI, All India Council for Technical Education, New Delhi, member of visiting committee of AICTE for the approval of institutions, member of scrutiny committee for approval of institutions, NAC expert committee member, CSIR extramural division expert member, and an expert member for NTA GPAT exam. He was elected as regional officer of AICTE Bangalore, Bhopal, Mumbai, Kanpur, and Kolkata. His awards and honors are: Sir has qualified GATE in the year 1986 conducted by IIT Kharagpur, received National Award for Best Research Paper of APTI in the year 2007 and Best Professor Award in 2019 by Devang Mehta National Awards. His national honors are: he was elected as President of Association of Pharmaceutical Teachers of India, Delhi from 2008 to 11. He conducted Northern Zone Conference for Development of Herbal Monograph in 2009. He was also elected as an Executive Committee member of IPA, Delhi. With this brief introduction, I now invite Professor Prabhat Kumar Sahu sir on board.
Sir, please unmute, sir.
Thank you, Deepika. A very good morning to all the dignitaries and participants of this faculty development program of AICTE. I respected Professor J. Surya Rao, Director of the Institute of Roland Institute of Pharmaceutical Sciences, and Chairman Professor J. Jayalakshmi, Professor Sabbu Thomas, Vice Chancellor of Mahatma Gandhi University, Kottayam, Professor B.V. Ravi Kumar, Principal of the Institute, Professor Niranjan Patra, Vice Principal of the Institute, and Professor Vishwamohan Sahu, the co-coordinator of this event, AICTE sponsored faculty development program, and all the faculty members of Roland Institute of Pharmaceutical Sciences, all the participants of different colleges all over India, and the faculty members and students attending this faculty development program conducted in Roland Institute of Pharmaceutical Sciences, sponsored by AICTE. I am very much delighted to be invited by the management of the institute, and I would highlight here that I'm a student of Professor J. Surya Rao, the director of the institute. He taught me anatomy, physiology, and health education in the year 1982 in Mahuda College of Pharmacy. So I'm so much happy, so very much delighted and very happy that I have been invited by such a great teacher who taught me, and because of his teachings and blessings, I could achieve different positions in AICTE, or whether it is Delhi Institute of Pharmaceutical Sciences or Delhi University. So always I seek his blessings. I am here very happy to see that AICTE has sponsored this faculty development program, and usually these programs are given to such institutes which are having very good repute, are accredited and approved by AICTE, and based on their performance and their reputation, this type of faculty development programs are projects are sanctioned to those institutions, and they carry out these programs to educate the faculty members in the field of pharmaceutical sciences in their respective areas. In Orissa, what I feel, this is the only college which is receiving continuously the grants from AICTE for faculty development programs and other research areas. I remember in 1998 also, those time I was in AICTE as assistant director. At that time also, I remember many AICTE grants were released to Roland Institute based on the quality of the institute, judged by various expert committee members. So here, since I have been invited to speak few words in this inaugural session, almost everything has been covered by our distinguished speaker, Professor Sabbu Thomas, about the quality of this faculty development program and the need of this faculty development program. I'll just highlight few words about our profession of pharmacy. This profession earlier, it was a neglected profession, what I can say, and during the year 1970, 1980s. Thereafter, gradually the profession emerged as a field of extensive research area. And whether it is ICMR, whether it is Indian Council of Industrial Research, whether it is biotechnology or whether it is UGC funds, all funds were going to the drug research areas. So I am observing since last 20-25 years, since I am associated with all these funding organizations, I observe that maximum grants is released to these drug development areas, whether it is cancer, whether it is Alzheimer, whether it is Parkinson's disease. So all these grants you will find is given to this research areas concerned to development of new drugs or development of new molecules. So our pharmacy profession, it has gained its reputation specifically during this pandemic area, a pandemic situation. That is, you see that whatever it may be, the entire world, entire Indian population, entire world, they are looking to a pharmaceutical house who will bring about certain new molecule or certain vaccine to save the population from this scenario of this COVID-19 scenario. So thereafter, everybody felt that this is an area which saves the entire mankind from this type of pandemic. So our profession, it gained its importance, and our this education, pharmacy education also, it gained its importance during the presidency of Professor B. Suresh. Sincerely, he is working to develop the curriculum, and ultimately, Pharmacy Council of India got its importance, and it to regulate the pharmacy profession. Earlier, it was regulated by All India Council for Technical Education. Then this Pharmacy Council of India, it was a dual authority regulating our profession. Ultimately, it was felt that yes, Pharmacy Council of India is sufficient to deal with the regulation of the pharmacy education, and it was given to Pharmacy Council of India. And during Professor B. Suresh has also developed all the regulation. It is pharmacy regulation, B Farm regulation, M Farm regulations, which we are not existing beforehand. They came into picture, pharmacy practice, all these new regulations came into picture during his tenure. And the number of institutions starting from few hundred, 10, 15 years back, now it has reached more than 2,000 institutions in all over India. Near about 1 lakh pharmacists we are producing every year, and they are serving the society. So our profession has gained this recognition, and you see the today, if you see what is Serum Institute of India and what is Bharat Biotechnology, all these organizations were not known to the general public. Even today, the supply, everybody is, every day you will find in newspaper that DS Remdesivir is not available, this, that, the wonder drug which is going to save the lives of the people. So this is our profession, and I am very happy that the profession was not, earlier it was not recognized, and people were feeling that yes, he is doing this pharmacy and he is a pharmacist. And but the time came that yes, the pharmacy profession and the pharmacist, the products which they are produced, that is going to save the entire mankind. So that is the importance of our pharmacy. And particularly in Orissa, I find the profession has also gained its importance. There are near about 20 degree pharmacy colleges and near about 60 diploma pharmacy colleges in Orissa. And this speaks about the growth and importance of our profession in our particular in our state, particularly in Orissa. And I am also very happy because I have visited all the nook and corners of the country because of my 10 years association in AICTE. And at many places I have found, wherever I visited, when I meet different expert members or senior professors, they speak about Roland Institute of Pharmaceutical Sciences. Yes, that there is an institution in Odisha which is very much reputed, very good infrastructure, and they are imparting very good education in Odisha. So these are the importance of the institute, and all the credit goes to our sir, Professor J. Surya Rao, who is himself he has all the entrepreneurial ability to bring up such good institutions in Odisha. And I hope the participants of different institutions who are enrolled for this faculty enrollment program will gain sufficient knowledge during the process, because there are a galaxy of reputed speakers in the panel, what I find, and that these this new education policy, that is education policy 2020, they have also given a lot of importance to this higher and technical education, and they have allowed different methods how to consolidate, and there are very much new systems they have developed, so that a person how he can get his degree and a diploma while doing his other courses also. These are the few highlights which will be discussed during this faculty development program. And I am very much happy that I could meet my teacher and a reputed speaker like Professor Sabu Thomas, the Vice Chancellor of such a reputed Gandhi University, Kottayam. And with this, I wish the organizers and all the participants that let the program be a very successful program, and let them go with certain value addition in the field of this pharmaceutical education. I thank the organizers and the principal and the management of the institution for inviting me to speak a few words before them. Thank you, all of you.
Thank you, sir, for sharing your valuable time with us. Thank you, sir.
Thank you.
Madam, unmute yourself. Unmute.
Thank you so much, sir, for enlightening us with your words. Now I request our beloved chairperson and patron of today's event, Srimati J. Jayalakshmi Madam, to present an e-certificate of next, we have another resource person of today's event, Dr. Vibhu Prasad Panda sir with us. Before starting the scientific session, I invite sir to come on board and share his view about this FDP.
Thank you, Ms. Deepika, for giving this opportunity. And am I audible?
Yes, sir. Audible, sir.
Okay. Thank you. The respected chief guest of the today's function, Professor P.K. Sahu, sir. Guest of honor, Professor S. Thomas, sir. Chairperson of the institution, J. Jayalakshmi Madam, and the director of the institution, J. Surya Rao, sir. Principal, Dr. B.B. Ravi Kumar, and the co-coordinators, Vishwa Mohan, and the other faculty members. Very good morning to one and all. And I'm Dr. Vibhu Prasad Panda from the School of Pharmacy, University, Malaysia. Just want to add a few points on this faculty development program. And first of all, I would like to congratulate the organizers and the staff members, faculty members, and the organizing institution, Roland Institute of Pharmaceutical Sciences, for organizing such a beautiful and the galaxy of the research personnel, not from India, all over the globe, and bringing into the one platform really is a commendable and need a note of praiseworthy. Really bringing into such a platform to all the research personnel in this COVID crisis, really it is a matter of pride to involve in these scientific sessions. So I will just I won't take much time because here just a few points I will to add on, because the research personals, the chief guest, the guest of honor already highlighted that what is the research and how to do the research. Just only one point I just like to restate or reintroduce, that collaborative research work is very essential at this moment. What we have seen for last 1 year is the COVID crisis, the pandemic crisis, and the successfully we have to come out with this challenge means only we have to get achieved by collaborative research work. So during my presentation, I will highlight how the collaborative research work has to be carried out, and I wish and I hope that during these 2 weeks programs of the faculty development program, the research personals, the faculties, and the participants will be highlight this part, how to do the collaborative research work and how to successfully come out to fight this COVID or fight with any unseen challenges what globally right now we are facing. So with this, I wish you all the participant delegates or the research professionals, the professional pharmacists who are involved in this program of 2 days program, 2 weeks faculty development program, going to be insightful, fruitful, and successful faculty development programs at Roland Institute of Pharmaceutical Sciences. With this, I take this opportunity to thank the organizers for giving me this opportunity, and hope see you all in my next sections of the presentation and this afternoon. Thank you all. Thank you for your time and giving this opportunity.
मैडम को लाइए.
Unmute.
Thank you so much, sir, for your enlightening words. Now, I request our beloved chairperson and patron of today's event, Srimati J. Jayalakshmi Madam, to present an e-certificate of appreciation to our guest of honor, Professor Sabu Thomas, sir, as a token of love and respect on behalf of entire Roland fraternity.
Sir, on behalf of the Roland Institute of Pharmaceutical Sciences, we present you this e-certificate. Please accept it, sir, from the team.
Thank you.
Yes.
I am very pleased and very happy to accept this. Thank you so much.
Thank you, sir.
Thank you, madam. I again request madam to present the appreciation certificate to our most respected chief guest, Professor Prabhat Kumar Sahu, sir.
Thank you, madam.
Sir, as a token of our love and affection, please accept this e-certificate for sparing your time, for sparing your knowledge for all of us. Thank you very much, sir.
Thank you, madam. Thank you, madam.
Thank you, sir.
Thank you, madam. Finally, I would like to take this opportunity on behalf of Roland Institute of Pharmaceutical Sciences to place on record our hearty thanks to Chief Guest Professor Prabhat Kumar Sahu, sir, and Guest of Honor Professor Sabu Thomas, sir, today's resource person, Professor Vibhu Prasad Panda, sir. We are really enlightened with your knowledge and presence. We are thankful to our Honorable Director, sir, and Chairperson, Madam, for constant support and motivation in all dimensions. I would like to thank our Principal, sir, and Coordinator of this FDP for his enthusiastic support. A special thanks to Co-Coordinator of this event, Dr. Viso Mohan Sahu, sir, for the timely arrangement and his precise efforts for the smooth conduction of the event. My special gratitude to all members of organizing committee, teaching and non-teaching staff members, eminent speakers from around the globe for sparing their valuable time, and all the delegates for their unflinching support and coordination. With these warm words and kind messages from our chief guest and guest of honor, we officially conclude the inaugural ceremony of Series 3 FDP. We will start the scientific session shortly. For this, I request Mr. Rajaram Das sir, Assistant Professor of RIPS, to start the scientific session and continue this event.
Thank you, ma'am.
Thank you, sir.
Good afternoon, dear all participants, and once again, welcome to the day one of Series 3 online FDP program sponsored by AICTE and organized by RIPS. Dear participants, today we have with us our first international speaker of this Series 3 FDP, Dr. Bibhu Prasad Panda sir, from the School of Pharmacy, Taylor's University, Malaysia. Before inviting Dr. Bibhu sir for his eminent talk, I would like to take the opportunity to introduce sir to all of us. Dr. Bibhu sir is an academician with a Doctor of Philosophy in the Pharmaceutical Technology, contributing to pharmacy education and research for more than 22 years in various capacities at national and international reputed institutions. He is actively involved in pharmaceutical and cosmeceutical product development and research. His area of research is mainly focused on nanopolymeric, nanocrystal, nanofiber, chronotherapeutic, and innovative drug delivery system. Till date, Sir has successfully guided many postgraduate and undergraduate students for their research dissertation work in the field of pharmaceutical sciences. Sir and his team has received various research grants such as Fundamental Research Grant Scheme from MOH Malaysia, MFS and ERFS of TRGS from the Taylor's University on design and development of nano-targeted drug delivery for cancer, Alzheimer's and diabetes therapeutics. Many of his peer-reviewed research and review papers has been published in high-impact journals, including Scientific Reports, Nanobiomedicine and Engineering, Micro and Nanosystems, Current Drug Delivery, Scientica Pharmaceutica, International Journal of Pharmaceutical Science Nanotech, and Pharmaceuticals MDPI, etc. Sir also serves as a reviewer for various journals including Scientica Pharmaceutica, International Journal of Biological Macromolecules, the Journal of Visualized Experiment, etc. Recently, Sir has received Best Presentation Award in First International Health and Medical Sciences Conference 2019 held at Malaysia for development of cosmeceutical research entitled Design and Development of Novel Transdermal Nanocream for the Delivery of Metformin. With this brief introduction, it's my pleasure to invite Dr. Bibhu Prasad Panda sir on board for his talk.
Sir, you make it hide, sir. Hide. Yes, yes. Hide, hide. Okay, right.
Am I audible? Am I visible?
Yes, sir. Audible and visible. You can continue, sir.
Okay. Thank you, Mr. Raja, and for your kind introduction and for presenting my brief profile to this August gathering. I am very delighted to know that more than 50 pharmacy professionals from all over the India are live for this faculty development program organized by Roland Institute of Pharmaceutical Sciences, sponsored by AICTE New Delhi. Good afternoon, my respected teachers, my dear professional colleagues, and ladies and gentlemen. I would like to quickly introduce myself. I am Dr. Bibhu Prasad, a pharmaceutical technologist cum academician from the School of Pharmacy, Taylor's University, Malaysia, and a former faculty and alumni of Roland Institute of Pharmaceutical Sciences. Heartiest welcome you all to today's my presentation entitled The Smarter Polymeric Nanocrystal Technology, Promising Platform for Design, Development, Delivery of Non-Communicable Therapeutics. At the outset, I would like to thank the coordinators of this faculty development program, Professor B.B. Ravi Kumar, Dr. Bishwa Mohan, all the faculty members, and the organizing institutions, Roland Institute of Pharmaceutical Sciences and AICTE, for providing this opportunity and the platform to present our research and research finding to this August gathering. This third presentation, we will present our overview of the research and in terms of a smarter polymer nanocrystals and its design delivery of non-communicable disease. So now I would like to start my presentation, and without further ado, because let me discuss straightway to my presentation, the smarter polymeric nanocrystal technology, promising platform for the design, development, delivery of non-communicable therapeutics. May I request Dr. Bishwa, can you show the slides? Now my slide is visible?
Yes, sir. Visible. You can continue, sir.
Okay, thank you for your confirmation. So let me start with my presentation. In this short presentation, as I said, I'm going to cover the second important points related to the sustainable development goals, new non-communicable disease, what is the research background, how to start a nanotechnology and nanocrystal technology, how to make a smarter polymeric nanocrystals, what are our research area, and how to achieve a research methodology, what adapt to develop a nanocrystal smarter delivery systems. And at the end, I will highlight how we have achieved certain points and contributed to this non-communicable disease. And last but not the least, my research collaborators and how to do a collaborative research work to achieve a successful in a non-communicable disease and its then management. So starting with the reason why, how to involve in this research, why to have taken this non-communicable disease, this is the story behind what we are looking into. The first point we would like to inform you that in 2015, in a United Nations, the General Assembly, there is a sustainable development goals and the global goals related to a collected 17 interlinked standards has introduced, and they come out with a blueprint to achieve a better and more sustainable future for all. So all the signatories of this UNO has now are responsible to achieve all these 17 points by end of 2030. So all the nations are trying to achieve these standards and the goal set by UNO by 2030. So in connection to that, the SDG 3, we have a 17 SDG. As we belongs to healthcare professional, it is our responsibility to look into the healthcare and well-being of the society. And every nation, every country is looking in that direction, how to achieve the good health and well-being by 2030. So being a healthcare professional, being a part of healthcare professional, it is our responsibility along with the nation, along with the UNO, we have to come out with this solution for achieving of good health and well-being by 2030. So with that intention, with that aim and objective in the mind, we have started realizing, and every country has compelled with a committee to come out with a certain guidelines how to achieve by 2030. So this is how we have entered into this UNO Sustainable Development Goal 3, where the good health and well-being is to be ensured. So under this good health and well-being, ensuring healthy lives and promoting well-being for all ages at all stages of the life is a big challenge for not only every country, for every healthcare professionals. So here how to achieve a good health, and good health for all ages and all time is not that easy. So in that scenario, we have decided, and as there is a guideline from each country, they how to achieve this. And the first and foremost important point is that improving health through a fundamental molecular studies and research. To do that, we have come out with that to improve healthcare of the society, we have chosen, our group of the researchers chosen the non-communicable disease and their management. So I would like to restate my statement that how to enter to this research background. Then it is behind this research selection criteria is the SDG 3, the Sustainable Development Goal, the set by United Nations, good health and well-being by 2030 to achieve. It is our prime objective of each country and each healthcare professional to contribute to achieve that goal point. So with that background, research background, we have started looking into how to do the research in the non-communicable disease and their management. As you know that the non-communicable disease like diabetes, cardiovascular disease, cancer, chronic respiratory disease, Alzheimer's disease are the biggest cause of the death in the worldwide. And out of this, we have selected the research for the non-communicable disease and their managements are the type 2 diabetes, cancer, Alzheimer's, which is remain a major challenge in the globe because of this non-communicable diseases and the fatal rate. So we have selected those 3 diseases like diabetes, cancer, and Alzheimer's in our research and to achieve this UN's SDG 3. I was very sad to inform you that at present the non-communicable disease management is going depicted in this picture presentation. If you look into this the scenario, you will find in the boat 4 professionals are working, 2 professionals in one end, another 2 professionals are sitting in another end. The first 2 professionals who could be the physician or the clinician or a pharmacist who is struggling hard to manage the non-communicable disease as the boat has already going to sink because of the water entering to this boat. And they are struggling to manage this non-communicable disease. Whereas the other part of this boat, you will see the pharmacy professionals like drug delivery scientists, molecular biologists, new synthetic NCE chemist, and all they are sitting in other part of the boat. They are thinking that non-communicable disease is not our responsibility and we can save by sitting the other part of the boat. But this is not the real story. You can see that the time will come, the boat is going to sink. So before going to happen to that, now we have taken into a challenge that being the pharmaceutical scientist, being a drug development scientist, drug delivery scientist, or new chemical entity designing scientist, or molecular screening scientist, it is our equally responsibility to manage this boat before going to sink into the sea of this non-communicable disease. This is how the motive us to take the objective to develop this healthcare system of non-communicable disease and their management. And we started looking into this way. And the most important part, we have taken into a decision that how to contribute as a drug delivery scientist or a technologist, how we can contribute to this non-communicable disease, that the most and foremost important technology and the platform, what we understood is a nanotechnology. The nanotechnology is going to be answer and not going to be a new concept, but it's going to be a successful platform for many therapeutics already proven their establishment in this society or society in the market, in daylight of the market as a drug delivery system. So just I would like to highlight, it's not a new concept, but it's already people are aware of this technology. Now we are going to use this technology in our drug delivery. So here the nanoparticles or nano delivery system, as you know that the size does matter here. The size less than 1000 nanometer is considered to be nanoparticular delivery system. And just to say that how the size does make matters in the delivery system, I would like to give a comparative of the particles or the size of the particles. Like a head pin is 1 mm, where the pollen grain is a 20 micrometer, where the RBC is a 2.5 micrometer, whereas the carbon nanoparticles are less than 1000 or 2 nanometer, 200 nanometers. So here the size does matter, and because of the size, you will find there is a tremendous potential and tremendous opportunities will be lies in delivery of the drug. So here you can see that nanotechnology is basically we can classify organic and inorganic way. And being a pharmaceutical scientist or drug delivery scientist, I think many of you, all of us, is more familiar with this micellars, liposome, dendritic. It belongs to organic one, whereas the inorganic like carbon-based, carbon nanotube, metallics, or silica-based delivery system. Now we are going to discuss about the nanocrystals. So nanocrystals is nothing but a drug delivery systems of colloidal delivery system of nanonized or nano-sized particles of drug crystals of dimension less than 1000 nanometer subject to appropriate nanonizing method. In a 1990s, this concept was more popular just to improve the solubility of poorly soluble drug and, by the way, improving the bioavailability of poor soluble drug. And the first drug has come out with this nanocrystal technology in year of 2000. It belongs to Rapamunes by Weight. They have introduced this molecule and the drug came out with this concept of nanocrystals. Right now, more than 5 drugs are in nanocrystal technology in the market. Around more than 25 products are going through a last trial, a clinical trial, and shortly going to release to the market. And this is how the nanocrystal technology going to be. So here I will just take a little introductory this nanocrystal technology, because this nanocrystal technology, there is a lot of changes happening in compared to last 10 years to this present scenario. Nanocrystal technology has developed and going to be the future era of delivery system. Now I will just highlight this is the nanocrystal technology are considered to be a different generation. One is called first generation, second generation, and third generation. And the first generation is called nanocrystals. They are basically a carrier-free, means no carrier. The drug will be converted into nano range of crystals with a stabilizer. Is considered to be a first generation, whereas the functional nanocrystals are basically they are carrier-mediated drug delivery system or nanocrystal system. And the basic difference between the nanocrystal and functional nanocrystals, as I said, here this is going to be a carrier-mediated delivery system, and you can see over here this is how a carrier of second generation is going to be a polymeric carrier system where inside the nanocrystals stabilized by a stabilizer, where is considered to be a second generation. It may go for a passive targeting, whereas the third generations are carrier mediated drug delivery system, which is having a carrier and a polymeric carrier inside the nanoparticles, nano drug into nano range, nanocrystals, and followed with a stabilizers. And again, you will find a surface modification will be there for a active targeting. So I will discuss in detail about each generation with a short time. I'll give you a scenario how things going to be under the nanocrystal technology. The nanocrystal technology first generation, as I say, mostly they are the same drug converted into nano range and keeping the crystallinity into their intact. And particularly we provide the stabilizer coating over here 0.1 to 1% to get a stabilizing the nanocrystal. Whereas the second generation nanocrystals, as I said, is a carrier mediated, and that is how a polymeric carrier mediated drug delivery systems are used. And in our research, we use the PLGA, PLA, many specialized polymers as a carrier for delivery of the drug into a second generation functional nanocrystals. We have used this concept of second generation nanocrystals not only for the cancer, even also we targeted certain drug to the brain, certain drug targeted for lung circulation by using PLGA, PEG. And one simple example also, we have studied that anti-cancer molecules like paclitaxel loaded nanoparticles for a controlled sustained release for a prolonged period of time and passive targeting by second generation nanocrystals. So second generation nanocrystal can be achieved. As you know that there's anti-cancer drugs, then the copolymers, peptides, and its ligands going to be forms a micelles. So micelles as a drug delivery system considered to be a inside the nanocrystal, that is considered second generation functional nanocrystal. Similarly, the liposomes is not a new concept, but we are adding the drug in the nanocrystal form into the liposome for the delivery. Similarly, looking into the third generation, I told you that third generation nanocrystals are nothing but the same nanocrystals. How is going to be modified the surface of the carriers and the polymers. We are modifying the polymer surface with the specific targeting antigens or antibody reactive moiety so that it can be achieved at the targeting drug delivery system. So here more specific active targeting is considered to be a third generation functional nanocrystals. So still today there is a Initially, people are talking about the first generation nanocrystals. Nanocrystal means without a polymers, but now there is a tremendous change in the concept and ideology of the nanocrystal technology. Now we are in a second generation, third generation, where we can make a polymeric functional nanocrystals, and that could be a new concept of delivery of many therapeutics agent, and we call it is a smarter nanocrystal technology. So as I said, the second generation or third generation could be go for a targeting, and third generation basically we use the targeting of the drugs putting into the nanocrystals, and it can go for a tissue targeting by enhanced permeability and retention EPR effect. You can see over here the blood circulation. This is the normal cell, this is the cancer cell. The nanocrystal can enter through the loose circulation and penetrate through the permeability and retention inside the biological, inside the systemic circulation for a long time, and it can achieve the tissue targeting. Similarly, it could be possible to go for a active targeting where the nanocrystal polymers will be modified with the antigens, antibody reactive reagents, and so specific targeting can be achieved. That also can achieve the active targeting process and reaching to the cancer cells and also the endothelial cell and to the blood circulation. In a simpler way, I said that the drug and the nanocrystals targeted molecules will be come out from the systemic circulation, reach to the tumor cells. Once it get reached, then you know that this is the third generation. I said the specific antigen and antibody will be react, and they will enter into the cancer cell. And by that way, we can target to not only the cancer cell, any targeting delivery system can be achieved by using the nanocrystal technology. The beauty of this nanocrystal technology is that the nanocrystal drug will be converted into a nanocrystal form, and along with polymeric delivery system, which can modify the release, which can make a active targeting, which can make a passive targeting, and much of things will be changed by including into a functional polymeric nanocrystals. As just a information here, how this is going to be, this is the polymeric nanoparticles, and these nanoparticles will find in case of the cancers, you will find the leaky vasculatures, and because of that easily it can come out from the vasculatures of the endothelial cell and reach to the cancers. And that, by the way, the nanocrystals which having the surface modification, now you can see over here these nanocrystals containing the polymeric particles and having a specific antigen antibody reactive way, they can interact with that and they target to the cancer cell. This is a very simple concept, but it's going to have a tremendous potential in targeting drug delivery system. And once it's entered to the cancer cell with this antigen antibody reactive process, you will find now the drug released to cytoplasm, and apoptosis is going to happen. Apoptosis is nothing but the natural death of the cancer cells going to be happen because of this targeting delivery system. With this brief introductory to the nanocrystal technology, we come out with the understanding that yes, the future of the drug delivery in the non-communicable disease is the nanocrystal technology, but particularly functional nanocrystal technology. So then we started our research, how to proceed with this research work. So we have, as I said, we have selected few non-communicable diseases like diabetes, cancer, and Alzheimer. And we have started working on this particular therapeutics and disorders and this particular treatment of therapeutic agents and using for our nanocrystal technology concept. So as I already told you, we have group of scientists working in these directions, and we have come out with successfully in delivery system which completed certain projects in the diabetes, like development characterization of second generation nanocrystal delivery system for the glycoside, then development of novel anti-diabetic polymeric nanoparticle of metformin, and characterization of the glycoside nanocrystal suspension. This project already completed, and we have received a tremendous encouragement to think for the further. So now we are still working some other moieties of diabetes molecule for targeting the diabetes, and also we have working in a cancer and Alzheimer. Right now we have doing the research on the cancer nanoparticle nanocrystal technology, making a conjugation with a specific targeting and with the antibody antigen reaction and moiety targeting to a breast cancer or triple negative breast cancer. Also, we got a great funding agency supported this research work, and I will present you later about how the research funding we are going to do this research work. Similarly, also we got another big project grant in Alzheimer's, doing the drug combined with botanicals or natural product for targeting to the brain and doing the treatment in Alzheimer's. Again, in this process, I have used the polymeric nanocrystal technology for third generation to target the Alzheimer's. So in my simpler way of approach in making the delivery system is that how to start a delivery system. I always look into this way, that the biopharmaceutical classification system is not a new, everyone know this, the class 1, class 2, class 3, class 4 drug, where you can see that class 2 drug is a low solubility, high permeability, class 3 is high soluble, low permeable, and class 4 is low soluble and low permeable. Either case of the class 2, class 3, class 4, we can use this nanocrystal technology for delivery of any therapeutic agent, irrespective whether it is going to be a non-communicable or a communicable disease. So I look always into that way to segregate my drug and how to how to improve the delivery, either by improving the solubility or improving the permeability. So the nanocrystal technology having the beauty of having both the benefit is a double edged sword, like that. One way it can improve the solubility, and also it can improve the permeability because of the nature of the particulate size. So that's how we use in our research work. So my prime research objective in being a part of the formulations or drug delivery scientist, what I do is that reinvestigating the effect of functional nanocrystal of existing therapeutics of BCS class 2, class 3, class 4 drug in non-communicable disease. And we have a very clear-cut guidelines how to do. Being an industrial pharmacy or technologist, mine is not going to synthesize a molecule. Mine is going to be reinvestigating the existing safe molecule which are suffering because of certain reason. They are not successful, but they are very safe, but they are efficacious, but having some problem in management. So that I am doing the research, how to reinvestigate, redesign the drug delivery system of the existing molecule to such a way that it can see the re-way and re-look of the molecule can be introduced into the market. So that's the how we are doing, and particularly my group of the researchers, we are working in this direction. So we examining the impact of nanonization, how the impact of nanonization or nanocrystal delivery system, either targeting or all these things, we are comparatively watching or checking the molecule in a pre-drug or active ingredient API or NCE, new chemical entity, and how does it have an influence after converting into nanocrystal form in the delivery system and therapeutic efficacy. Is a great potential for all the boarding scientists, all the researchers can take this project, that you can do a tremendous research work, again redefining the drug into nanocrystal form, into first generation, second generation, third generation. You will find there is a lot of change in the delivery and therapeutic management of that particular active ingredients. So there is a lot of potential, can rethink about the reinvestigating the existing established molecules. We are not synthesizing a new molecule, but we are redefining, resynthesizing, and redelivering the drug as per the nanotechnology concept, using and redefining the management of non-communicable diseases. So our research outline is very simple. And just one is I'm highlighting because I told you we are working in the diabetes, cancer, and the Alzheimer's. But at present, we have successfully completed the project related to the diabetes. So in my research, how to proceed for any research, I'm just highlighting my example of doing this type 2 diabetes, non-communicable disease, how to proceed as a starting of a new researcher like me. I'm a new research into this field of non-communicable disease, how I have completed my research and citing with the diabetics and molecule. I will cite and give you an example how to proceed for this research work. So as I said, we are taking a oral hypoglycemic agent. I told you we are not synthesizing a new molecule. We are redefining the molecule, how to improve by delivering in a better way by using the nanotechnology or nanocrystal technology concept. We have used many drugs related to oral hypoglycemic agent. As you know, glycoside, metformin are well-established molecule in the diabetes. So we relook into that delivery challenges. What is the problem? Why that non-communicable disease in the diabetes still is not controlled? What is the problems associated with these molecules? So we started looking into that way and started using in our research. And to do any research, as you know, we have to start with the design of the experiment. We do a 3 factorial design or Taguchi orthogonal design, any experimental design you can adopt in your research work. So we have started with a glycoside is a one simple example. Here our objective is design, develop and characterize a stable, optimized second generation smarter nanocrystal drug delivery system which can modify the delivery. That's the objective and the aim, and enhance the drug solubility, enhance the dissolution, improve the bioavailability with low therapeutic dose of the glycoside. That's very important point to our objective is that how the dose can minimize because of nanoparticulate delivery system. Is it the same drug of the normal drug dose and compared to nanonized drug, how this drug efficacy also we are re-investigating. So this type of the work is actually very limited research work is published, and we are looking redefining the existing molecules. As I said, our methodology is a very straightforward. We started with the design of the experiment. So we use 3 to factorial design or Taguchi, then started doing the real after going to experimental design. We synthesize, we develop the delivery system by formulating a different approach we adopt to making of the nanocrystals. Simple example I can highlight here. We have a different approach, but both the approach we can successfully completed making of the nanocrystal like emulsion diffusion, high pressure homogenization and solvent evaporation technique, and another technique called nanoprecipitation technique and high pressure homogenization solvent evaporation technique. Then after making the formulations and optimization, we characterize in vitro characterization has to be carried out for the smarter polymeric nanocrystals like particle size distribution, then photon correlation spectroscopy, TEM, SEM, transmission electron microscopy, scanning electron microscopy, atomic force microscopy, zeta potential, solubility, in vitro drug release, entrapment efficiency. Similarly, we will look into the drug excipient compatibility with the DAC and FTIR XID studies. Later, once you will confirm this in vitro drug delivery systems is stable and is going to be a promising, then we will go for further characterization for in vivo characterization, where we do some animal study and we develop the formulation up to a preclinical study. Not we have done any clinical study, and this is under the trial. We planning for the future work, but not yet we have done a clinical study, but we are bringing the molecule up to a clinical study that's called preclinical study using a in vivo characterization by using a animal model. We have come out with the pharmacokinetic, pharmacodynamic studies, bioavailability study, and pharmacokinetic, pharmacodynamic correlation studies for establishing of this concept of delivery of nanocrystal in the non-communicable disease. Finally, we proved that our delivery system or developed formulation is stable and stability as per the stability guidelines. I'm not going in detail, just I want to highlight you that we are started with the designing of the experiment. As I say, we can use the design experiment softwares. They will provide you with the independent variables. We use PLGA, polaxomer, and dependent variables like particle size, drug release, all will be used. And by putting this into the design experiment, they will propose you a set of the experiment, how to conduct, and this is how the experimental design will provide you guideline to do the experiment as per these guidelines. So we come out with those experimental design into our lab, and then we have carried out the formulation development. Similarly, a Taguchi experimental design also can be adopted, where you can see here the independent variables and with the 3 different levels. Then the software provide you the guideline how to go for the combination in a short time in a scientific way. That is called Taguchi experimental design. And the Taguchi experimental design provide you a guidelines to make the 9 different formulations, and then we come out with this combination in our lab to define and develop a formulations. So once based on the formulation designed by the software, then that we have put into our lab to develop the formulation. I told you we are approaching a 2 different approach. One is called EH technique and NHS technique. emulsion diffusion, high pressure homogenization, and nanoprecipitation technique. Then by this both the process, we develop the second generation or third generation nanocrystals. After developing these nanocrystals, we go for the characterization. Already I told you, in vitro characterization and in vivo characterization process. So in a simpler way, how we develop this particle, this drug delivery system in the lab, in a simpler way, we develop with emulsion diffusion high pressure homogenization technique, where the PLGA is ideal with many different polymers, but the most preferable one in my research is PLGA, polylactic glycolide is the most preferable one. And we do with the glycolide many drugs of non-communicable disease. Then we mix with the aqueous phase with a different stabilizer, then go for the homogenization, followed with the formation of the emulsification, first phase emulsification. Then we go for high pressure homogenization and overnight solvent evaporation. And by the way, we will get a nanoparticle in a suspension form, and sometimes we convert this nanoparticle suspension into a freeze-dried powders with lyophilization technique. So similarly, another technique also we do nanoprecipitation. It's the almost same, only there is a different process of making, is the releasing the this process of starting stage by using a mechanical or manual way, allowing the drug and making a nanoprecipitation technique, and then later we make high pressure homogenization followed with solvent evaporation techniques to get the nanocrystals. Both the method we are successfully achieved the first generation, second generation, and third generation nanocrystals. And later, after developing the formulations, now we do the in vitro characterizations. We use zeta sizes and we analyze the particle size, and we see the potential nanoparticles of first, second, third generation. And we analyze the particle size, polydispersity index, and zeta potential, which are the prime objective in characterization of the nanoparticles in any delivery system related to nanotechnology. We do all this, as you know, most of you know this. Polydispersity index is the dispersibility, monodispersibility is the dispersibility less than 0.5 is considered to be an ideal one, and we achieve 0.02, and the particles is 100 around nanoparticle size. Zeta potential, good negative zeta potential help you a stable nanoformulation. Then we do the stability studies and solubility study in a different solvent systems like phosphate buffer, 0.1 HCl, and water, and we check that how the nanocrystal has improved their solubility. Then after making this confirmation, we go for the characterization by field emission FESM studies called scanning electron microscopic, and we examine what particle size we analyze with the zeta sizer, reconfirm with this scanning electron microscopic method. We can see here, this is the placebo, means without addition of the drug, the particle size, which is reported by zeta sizer, the particle size, and this is the drug loaded nanoparticles, and it is proved that the emission microscopic also shows the surface morphology and the particle size. It's further confirmed by doing the TEM study, the trans-electron microscopic study, to confirm the particle size. What we claim is further confirmed with the zeta sizes, the particle size, and this is the placebo without the drug loading. This is with drug loading, and this you can see the confirmation that nanocrystal will be confirmed with these studies in vitro surface morphology and particle size analysis study. Further, we confirm this delivery system by doing atomic force microscopic studies and of the placebo in 2D form and 3D form, and it indicates that a very spherical structure is achieved, the nanoparticles. That is the indication of how the nanocrystals, polymeric nanocrystals or functional nanocrystals can be looks surface morphology, and this is the confirmation of that. Further, we do that the nanocrystal technology is assume that drug after converting the nanocrystal form, is the crystallinity still retained or not? To prove that, we do XRD, and we see the pure drug and the optimized formulation. Still, we find a specific sharp peak indicate that the nanocrystal properties of the drug is not altered. Further, we confirm with the drug excipient compatibility with the DAC studies or the pure glycoside and the nanoform optimized formulation. You can see over here the sharp peak indication that the nanocrystal optimized formulation still it retained the its crystallinity and the integrity of the molecule. Further, it's confirmed with the FTIR studies that the characteristic speaks of the pure drug and optimized formulations still retain, and means it is the integrity of the molecules is further confirmed in the in vitro studies in examination that yes, the drug is capable to deliver into the nanocrystal formulation. This is the proof help us to take further to our study to go for the drug release study. And now we can see the drug release of the pure drug and the nanocrystal form, and you will find there is a tremendous change in the release profile, and this is also confirmed with our in vivo pharmacokinetic studies. So after taking this data, we will put into the software, the experimental design software, and by using this statistical design softwares like experimental design or Taguchi experimental design, they will predict a set of the experiment optimized formulation, and you can use the contour plot, and all these things can use for determination which way the formulation proceed to get a optimized formulations. So this similarly, the distribution plot is a statistical analysis how to achieve optimized formulations. So once we confirm our in vitro studies, then is the promising molecule of the drug of NCD into the delivery system will be exposed to the in vivo studies with animal studies. So we, in this example, we have induced the diabetes for the anti-diabetic molecule. We have used for oral hypoglycemic agent, and we develop a model by using a streptozocin nicotinamide model for inducing the diabetes to the animal. Then we studied our nanocrystal, how promising it is. Then with that, we started doing the pharmacodynamic studies like the blood glucose level, how reduce. You can see over here the placebo means without the drug, this is the placebo. The blood glucose decrease compared to that time. You can decrease of the time. You can see here the pure drug is this one, and the nanocrystal form, there is a drastic change in the blood glucose level and bring down into such a way this is highlighted by our nanocrystal technology in the blood glucose control of non-communicable disease in type 2 diabetes. Similarly, we have done the pharmacokinetic studies. We found the plasma glycoside concentration and versus the time profile. This is the pure drug. This is the nanocrystal second generation drug delivery system what we have developed. It's clearly indicating there is a tremendous change in the delivery of this nanocrystal delivery of glycoside. Then we have done the pharmacokinetic pharmacodynamic correlation study, and there is a good correlation was achieved, and which you can prove that the pharmacokinetic and dynamic are equal correlated, means as the drug concentration in the drug in the plasma will be increased, there is a pharmacodynamic response equally responsible, means the drug is releasing from the delivery system and going to the receptor level and stimulating the insulins to control the blood glucose is established by this pharmacokinetic pharmacodynamic model. So we come out with a comparative way to see that how the pure glycoside and optimized nanocrystals has improved. You can see over here the simple important point to note that here the area under the curve indicate the bioavailability is a 5.3 for a pure glycoside, whereas the optimized formulation has a 6-fold improved in their bioavailability. Similarly, you can see the biological half-life in the case of pure glycoside is a 9.75 hours, whereas the 29.56 hour the drug will be remain in the body. So that is the great findings in our delivery system of nanocrystal, that you can minimize the frequency of administration of the drug because the drug is remaining in the blood for circulation for a long time. That could be used for a long circulating delivery system, which considered to be a passive targeting. So these are the promising findings we have noticed, and after that we have carried out the stability study. Also, we proved that our drug delivery system is stable in a short-term stability study as per the guidelines. So we come out with the understanding that this delivery system, what we use, the smarter polymeric nanocrystal system, particularly I'm highlighting for this oral hypoglycemic agent, what we have completed the research highlighting our findings. So these nanoparticles are good, having good zeta potential, good polydispersity, low polydispersity, high entrapment efficiency, improve the solubility and drug release correlated with the in vivo performance. you can prove it can seen from the drug release profile and also in the in vivo pharmacokinetic study. So from this delivery system, we able to achieve a better bioavailability and therapeutic efficacy. And we have noticed that this type of the PLGA based nanocrystal second generation drug delivery system able to produce a biphasic release profile of the polymeric nanocrystals. And this is also confirmed that initially quick release in the in vitro study and also proved in the pharmacokinetic study that initially the drug release very fast, then followed with a delayed release profile. So this is how is the beauty of this PLG nanocrystal technology, and that is what we call is a smarter delivery system. And it's a just finding we have noticed that this unique pattern of the drug release of PLGA based polymeric nanocrystal in the oral hypoglycemic agent can be explored to meet the typical physiological need of type 2 diabetes. That's the most important findings in our research, that in a type 2 diabetes, what we have seen that after taking the food, the blood glucose will be increased and then will be remain. So this type of the delivery system, if you administered along with the food, then you will find it will be initially release the blood glucose, decrease the blood glucose by releasing the drug, then followed for 24 hours to 29 hours, remaining in the constant level, what is desirable. Because after taking the food, the blood glucose will be suddenly shut off, which can be minimized by giving this type of delivery system. So that is the how this delivery system considered to be a smarter delivery system. We define and recoin this delivery system as a smarter delivery system because the faster drug release at the time of the meals followed by a prolonged drug release profile over an extended period of time to maintain the constant plasma glucose level, which is highly desirable, and better patient compliance and better drug therapy in the non-communicable disease of diabetes type 2 can be achieved. So this is what we have produced and published and we have achieved also. So as I want to highlight you, because of the shortage of time, I'm not going in details. Just let me tell you in a brief collaborative research work. I told you our research work is a collaborative research work. We have received very good research funding from different countries, from different parts, and from the Malaysia, from different agencies. So recently we have received one research for 32 lakhs Indian currency, INR, 32 lakhs, more than 32 lakhs, for the research of investigating a anti-cancer activity, anti-MIRA 21, MIR145, mimicking the plus doxorubicin for cocktail package of nanoparticle conjugated with A6 tumor homing peptides for targeting to the triple negative breast cancer. So this is the most advanced delivery system we are developing with using same concept of targeting nanocrystal technology. We are using a homing peptides, A6 peptide, conjugating with some mimicking anti-cancer mimicking drugs, doxorubicin, along with some anti-MIRA 21 for targeting a triple negative breast cancer. So this is the research work we have received recently, and we are working, and the work has started last year. Similarly, as I said earlier, we have also received 78 lakhs the grant amount for making a neurological disorders for the prevention of the neurological disorder. Particularly, we are working for Alzheimer's disease in case of the anti-aging for aging populations. For this non-communicable disease, we have used basically a natural plant compounds along with a certain drugs, and now we are making the delivery of the nanocrystal technology and using the natural plant ingredients and looking into the degeneration of the beta amyloid protein, how it's going to modify by this nanocrystal technology and delivering of the natural ingredients. This research work also started. It is on the halfway, and we have got a very promising result so far. This is how we are working in presently we are working in the neurological disorder, particularly Alzheimer's non-communicable disease. And major schemes, already I told you, we have already completed the 3 research projects. related to the diabetes, particular type 2 diabetes, and already completed this research, like design development of second generation nanocrystals. And we are, and particularly I am this drug delivery part, I am particularly working, and this as a principal investigator of this particular projects, and we have completed 3 successful projects. And similarly, we have developed nanoparticle polymeric and nanoparticle for metformin delivery system. It is also completed, and we have published few research findings from the present research work. And recently, we have published a good research work and in the scientific reports of Nature's Journal, and called Fabrication of Second Generation Nanocrystal Smarter PLGA based nanocrystal carrier for improvement of drug delivery and therapeutic efficacy of glycoside in type 2 diabetes rate model. So this is the, and recently we published, but it was acclaimed and throughout the highly encouraged by different platform. And this is the really good work. Everyone is appreciated. Really, it encourages us to take further study in the nanocrystal technology. And now we are working into other fields of non-communicable diseases also. We published many related articles, publications like new therapeutic approaches towards management of diabetes. Then we published few papers like influence of the stabilizer, how to develop a second generation nanocrystals. Also, we published a statistical design, as I said, the experimental design, how can you use in the design of the experiment of nanocrystal formulation development. And that is also we published in recently some publications we have done. And these are some papers, few related papers related to second generation, third generation also we publish. And we also publish some related to the current trends in design and development of nanocrystal nanopharmaceutical dosage form as a review articles. So these are the few research paper we related to the research project I'm highlighting here. So as I said, this is the time we have to think a collaborative research. Going out of the in silo means doing the research self, and definitely we are capable, but it's not going to give a good result unless we go for a collaborative research work. So that's what I understood within my few years of my research entering to the nanocrystal technology delivery system. I understood that it's not only the technologist, because we need a clinical pharmacokineticians, we need a molecular biologist, we need many scientific people for their contribution to develop a better delivery system. So at present, right now we have 3 research scholars, Ms. Puna, Ms. Jaida, Ms. Rachna is working under this research work of non-communicable disease. And we have a great collaborators like Prof. Kates. You know, he is Prof. Kates is the director of Institute of Green Nanotechnology, director of University of Missouri Cancer Nanotechnology Platform, Columbia, USA. And most of you know that he is considered to be a father of green nanotechnology. He is a collaborator with our research work, and we are working jointly in our nanocrystal technology drug delivery system. Similarly, the Professor Chong P is a molecular biologist and microbiologist from the School of Bioscience. Dr. Priya is the Taylor's University School of Medicine, the clinical pharmacokinetics and the clinicians. Similarly, Dr. Subrat is a pharmacologist from the University of International Medical University. He is also a collaborator. Dr. Naveen is the pharmacist, the pharmaceutical chemist. Dr. Bin Singh is the School of Medicine, is also molecular biologist and clinical pharmacokineticians. Similarly, Dr. Lee is going to be a bioscience, and the mechanistic establishing of this molecular delivery system, she is looking after of our research project. So what I just want to tell you is that the research is not going to be a single man's story and not going to be a successfully. If you want to see a successful story, then definitely you need a good collaboration and quick getting a result, good publication, good for patents to go, then you need a good collaborations. So that's the reason I have highlighted my collaborators and co-researchers to show you that the research is not a single man show or single man studies of a technologist or single way a new chemical entity. If you synthesize, simply you can say that yes, my delivery system is upright. No, you have to go for a collaborative research work to make a successful delivery system. So I acknowledge the School of Pharmacy, Taylor's University, Malaysia, for providing and this all these facilities and encouraging me to just like I'm a simple beginners of the researchers. I'm just learning, but I learn how to do collaborative research work in this from the School of Pharmacy and how to collaborate. That's what I learned. Though I am having a 22 years of teaching experience and research experience, but I have learned last couple of years that how to do collaboration and how to do the research to take next level of my research journey. Also, I thank the Roland Institute of Pharmaceutical Sciences and AICTE for providing this opportunity to give this platform to share my research and the what humble way we have done to this non-communicable disease I have just presented over you. So if you have any question, please feel free. I am happy to answer your queries if you have any. Now I open the to have any discussion, any queries. What is the way I can able to answer? I will glad or happy to answer your queries if you have any. So Dr. Bishwa, I can open the floor now for the participants or the research scholars or anyone who will be here to discuss. Can I able to see what the question, because I'm unable to see anything in my screen.
Yes, sir. Yes, sir. I have received some questions, some chat box. I'll display on the screen, sir.
Okay, please, please. Thank you.
Is it visible, sir?
No, still I am not able to see. I am in the slides only. Thank you, slide. Last slide.
You can minimize, sir. You can stop that one.
Okay. Okay. Yeah. Okay. Thank you. It's a great question. That somebody is asking that what is the safety concern of the nanocrystal formulations? That's the reason I told you the nanocrystal technology or nanotechnology having a tremendous potential for the research. We can just consider the drug and converting into nanocrystal, you can consider as a new drug because the characteristics is going to make a tremendous change in the delivery system as far the nanotechnology is concerned, whether you talk about the nanocrystal, whether you talk about the second generation or third generation, the nanocrystal or nanotechnology itself is having a tremendous potential, not only in therapeutic efficacy, but also going to be in the toxicity. So there's a great potential to go for the research, and many research has not done the toxicity and safety profile of nanoparticles or nanotechnology based drug delivery system. So this is a open space for the active researchers in the nanotechnology can adopt this concept and can do the study how to establish how the drug of the pure drug compound and how the drug of nanocrystal and their impact in therapeutic efficacy and safety profile also to be a great potential to do the research. Yes, there is a change in the safety profile and we have to do the safety profile study also. So the next question is, good afternoon, happy to listen your talk after a long time. What are the advantages of nanocrystal over a nanovesicular structures or nanocrystal sustain release better than nanovesicles? So the first and foremost important thing is that we have to understand that nanotechnology is almost having the nanonizing of the drug delivery system. So either nanocrystal or nanovesicular structure, you are bringing down the particles or delivery system to nanonizing. So because of the nanonizing, then there is going to be a better permeation, better bioavailability. That is one part, irrespective of what delivery system you related to select for the nanotechnology. And the beauty and the importance, significance of the nanocrystal technology is that the nanocrystal technology is going to retain its integrity. So you can see that crystallinity property of the drug is not going to be compromised. So that's the reason. Another way you can see that nanocrystals second generation, third generations are considered to be a much advanced delivery system compared to a vesicular structure. In that you can see that the drug is not going to change, but most of the vesicular nanostructures you delivery system, we are modifying the drug, and when you go for the nanonization, you will find most of the cases the drug is going to be converted into a crystalline into amorphosic form, which is going to change major changes in physicochemical properties, and that has to establish the therapeutic efficacy. So that's the reason nanocrystal is considered to be an ideal delivery system in nanotechnology delivery system among all these nano delivery systems, that the drug integrity, physical, chemical, therapeutic efficacy is not going to change because the physicochemical properties of the drug will be retained inside the delivery system. And other than that, second generation, third generation, can you make it for a passive targeting or a active targeting? Hope I have answered your queries.
Okay, thank you, sir, for your nice presentation. So it will be very useful for the faculty members and also research scholars who are involved in the recent technology, that is nanotechnology. It will be very useful, sir. Thank you, sir, for your valuable time.
Is there any other further questions?
No, sir. This is up to this we have this.
Okay, then it's a great pleasure being with you all in this scientific session. And thank you all for your time and the patience hearing of my presentation. I wish you all a insightful and fruitful 2 weeks faculty development program, and wishing you all a pleasant day and the prosperous happy new year 2021, and goodbye and good luck, and wish you all a successful 2 weeks faculty development program to the organizer and to the participants. Thank you all for your time and patience hearing, and thank you all once again to the organizer for giving me this opportunity.
Thank you, sir.
Thank you.
Thank you so much, Dr. Bibu Prasad Panda sir, for taking such an innovative and scientific step towards improving the healthcare of modern society through this smarter polymeric nanocrystal technology targeting the major non-communicable diseases worldwide. May I now request Professor BVV Ravikumar sir, coordinator of this program, to present an e-certificate of appreciation to Dr. Panda sir as a symbol of our love and affection from entire RIPS fraternity.
Thank you all.
Thank you, Professor Bibu, for accepting our invitation and coming online to give useful information.
It's a great pleasure being with you all, to my alumni, to my Roland Institute of Pharmaceutical Sciences. I feel a great honor and privilege to being associated with my alumni and the Roland Institute of Pharmaceutical Sciences, where I've started my research career, where I've started my academic careers. So I'm very grateful to you, sir, for giving me this opportunity and inviting me to this platform. Thank you very much.
Thank you, sir. Please accept this e-certificate of appreciation on behalf of Roland Institute of Pharmaceutical Sciences.
Thank you very much, Professor B.V. Ravikumar and Vishwa Mohan, other faculty members, and I've accepted open heart your certificate of appreciation you have given to me, and I wish you should send to me through my online mail ID. So that will be much useful than receiving this on way. Thank you very much.
Thank you, sir.
Thank you, sir. Thank you for sparing our time.
What is the secret of your education smartness?
So I think you know much better than any other participant, because we have a very good association for last more than 10 years and 20 years. So don't open the secret in front of the others. So please keep it in the secret. That the secret is that the love and affection what you are giving is really makes me to younger and active in this research work. Thank you very much for your all support and guidance, and your love and affections really appreciable.
Thank you, sir. Bye.
Thank you all. for your support and
Thank you, sir.
encouragement.
Thank you, sir.
Okay, so I'll stop over here.
With this, the day one of Series 3 online FTP program is officially adjourned. Every end is the indication of a new beginning. So we will be starting the day 2 of this online FTP program tomorrow morning at sharp 11 a.m. on the same platform. Till then, please stay safe and stay connected with us. Thank you all the participants for your kind attention and your participation. Thank you very much.